drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. This episode is ad-free, and I rely on my audience to support my efforts. If you're getting value from this content, please consider becoming a paid subscriber to the Mind and Matter Substack. For just $5 a month, you'll get early access to episodes and other content, full episode transcripts, and I'll prioritize answering your questions in emails and the comments sections. You can also support me by checking out the links in the episode description to my affiliate partners who make various health and wellness products that I use and enjoy myself. You'll receive a discount and help support the podcast that way. And if you want the benefits of a paid subscription, but it's not in your budget, simply sign up for my free weekly newsletter, send me an email, and I'll give you a complimentary paid subscription. Word of mouth is the best way to help the podcast grow. So if you like what I'm doing, please share your favorite episodes with family and friends. The purpose of the podcast is to provide you with information obtained largely from primary sources, the people doing the research and producing new knowledge. This content is never meant to serve as medical advice. And with that, enjoy the episode.
Thanks for having me, Nick. Appreciate it.
Can you start off by just telling everyone a little bit about who you are and what your lab studies?
Sure. So I'm at the University of Utah. I'm in the Division of Microbiology and Immunology in the Department of Pathology. And my lab is interested in the interaction between the commensal microbes that live on your body and the immune system. And we're really interested in how these organisms promote health. So I think a lot of people, when they think about microbes, they think about disease, they think about pathogenesis. Of course, they equate microbes to the pandemic. They're always just thinking about getting rid of microbes. But we think about microbes in a very different way. We think that there's a lot of microbes that are present on a healthy body. And our real interest is identifying those microbes and how we might be able to utilize them to prevent diseases.
Mm-hmm. And most people, when you start talking about the microbiome, people talk about good versus bad bacteria. It's intuitive as to why they're doing that, right? Bad bacteria would be, a pathogen, something that could make you sick or even kill you potentially. A good bacteria, I guess what's implied there is that it's contributing something to your health or to the functioning of the organism, potentially even something that's necessary. Do you think it makes sense to talk about good versus bad bacteria? And if so, what does good mean to you exactly?
It's such a good question, Nick. I guess I can't help but think of good versus bad because a lot of our studies have identified bacteria that are beneficial. But I think in reality, it's definitely more of a spectrum. Good bugs can go bad, for sure. There can be organisms in your body that might be providing you a benefit. And then if they're provided an opportunity, let's say they get out of the gut and into the systemic part of your body, they could cause a problem. So I think what you're hinting at is absolutely right. And there's not always good bugs and always bad bugs.
Mm-hmm. So some of the good ones can become bad. And my understanding is that this often means, they have to sort of be in the right place. They have to be contained in the right area. And if they get out of where they're supposed to be, quote unquote, then, as you said, they can become bad.
That's right. Yeah, definitely they need to be contained. Most of our microbiome lives in the intestine. So certainly if those microbes get out, let's say they get in your liver or in your blood, they're going to cause problems. Although, most of them are highly anaerobic. So they, can't survive in highly oxygenated areas. Mm-hmm. So most of them will rapidly die. But yes, if they get out of the compartment, they can do some bad things.
Yeah, and I've had microbiome people on before, and we've talked about various aspects of it. So my understanding is basically, right, we've got our immune system, you've got microbes in your gut and elsewhere. And the immune system is there's sort of a delicate balance there. On the one hand, you don't want to destroy some of these microbes, but on the other hand, you want to contain them, keep them at bay, prevent them from going where they're not supposed to be. Can you give us just a general basic sense for some of the communication that happens between the immune system and the microbiome?
Sure, definitely. Um, I'll focus, I guess, on the things that my lab studies because I know that stuff best. So we're really interested in antibodies. So most people are familiar with what antibodies are, but they're molecules that B cells produce. And you make a ton of them in your gut. The average human makes about 2 grams a day of an antibody called IgA. And these antibodies are one of the primary ways in which we contain our microbiome. So these antibodies are very specific for microbes. There's lots of different parts of your immune system. Some of your immune system is very general and just recognizes, cell wall components of bacteria. But there's another part of your immune system which is very specific. And this is the part that antibodies are part of. So these antibodies are highly specific for the microbes in your gut, and they help to control them. And they don't just, they do keep them contained in their area, like we were just talking about, but they also control the types of molecules that they let those bacteria express. Bacteria or fungi are viruses, I should point out. So they don't let organisms express molecules that are harmful to the host. So if there is a harmful toxin or adhesion or something that makes that organism, invasive to the host, antibodies will target it, and that bacteria will downregulate expression of it. So it's not about always getting rid of the bacteria. It's kind of about controlling their function, their behavior.
I see. So it sounds like we IgA, this antibody we produce quite a lot of it. And if I'm hearing you correctly, normally when I sort of naively think about antibodies, you sort of mass produce them when you get sick, and they sort of just cover the entire outside portion of a bug, and then that's sort of a step towards destroying that bug. But it sounds like these IgA antibodies, they can actually be used not to sort of destroy or completely suffocate these microbes, but they actually just control there's sort of a molecular communication there, and it controls what the pathogens are actually making molecularly.
Yeah, that's a great interpretation of it. And most people think like you, Nick, where they think about the mass production of antibodies and they're in our blood, and their whole function is to tag a microbe for destruction. And IgA does do that in the gut. It just, IgA is different in the sense from other antibodies in the blood because it doesn't mount, there's no inflammatory response associated with IgA. Its sole purpose is to target those organisms, and it does get rid of, basically it groups the organisms that it wants to get rid of together, and then you will essentially poop it out. So there is some eradication that happens, but ultimately those bacteria, because of that response, will downregulate those molecules.
And before we get into some of the specifics around some of the work that you've done, some of your recent studies, so when we say that there's good bacteria, and with the caveat that good bacteria could become bad, and things are a spectrum, like what are some of the major things that we know good bacteria generally do? Is it that they are providing key nutrients that we can't really produce otherwise or absorb otherwise? Is it that they're sort of merely taking up space to box out pathogens? Like what are some of the good things that tend to come from these gut microbes?
Well, I think you named, some of the top 2 that people think about is, first of all, they take up a ton of space. That kind of excludes any pathogenic bacteria. This is actually like a really important function of the microbiota because they have evolved to live with us peacefully, whereas pathogens haven't. So by them taking up the space, that prevents us from getting sick. And C. difficile is probably one of the best examples. So if you undergo antibiotic treatment, you kill a lot of those commensal microbes, and then C. diff has an opportunity to bloom. And this causes a lot of people problems. Another major benefit of bacteria is indeed metabolism. So they do, we can't process a lot of the fibers that we eat, like cellulose in plants.
Hmm.
Our microbes help us extract energy in things that we might not normally extract. They also produce molecules that help absorption of certain things like lipids and sugars. They can also prevent absorption of things that perhaps are not good for us. We still don't understand fully their metabolic capabilities, but we know that they're really important for metabolism. And then, of course, one of the major things, and that's what we study in my lab, is that they're really important in teaching your immune system about what to fight off and how to fight it off.
Hmm.
So in our lab, we use germ-free mice, which maybe you've had previous guests talk about, but these are mice that have no microbiome of their own. And if you infect these animals with any sort of pathogen, they basically just die and it's largely because their immune system hasn't been taught how to fight off that pathogen. So it's like really important for our immune system maturation.
Yeah, so the immune system has to be trained across development in order to function properly and effectively at getting rid of pathogens. You're saying if you create animals that don't have a microbiome, basically, there's no bacteria living inside of them or other microbes, their immune systems just don't learn how to function properly.
That's right. Yeah. So if you compare their immune system to that of a normal animal with a normal microbiota, they're missing a lot of immune cell populations.
Hmm.
Or the immune system populations that are there are very naive, or they're, reduced in number. So it's a really important thing our microbiota does, is to teach our immune system.
And when you say germ-free animals, how are you making these? Is this like a genetic thing, or do you have to give them lifelong, antibiotic treatment from birth?
So so we don't antibiotic treat these germ-free animals. They basically live in these little bubbles.
Hmm.
And you have to be very careful about the food you feed them, and we just have to keep them in the bubbles and only give them, sterile food. The way the original germ-free animal was created was that you had to do a C-section of a pregnant mouse mother.
Mm-hmm.
And then those babies, they're like beads on a string. So you basically plop them in a little sterile solution, and then you place them in this bubble.
Hmm.
And then you take them out of their sac, and someone had to hand feed the first germ-free mice ever made, because there was no mothers in the.
Wow. So a couple of things here, like this is just sort of a lineage that's been going forever like this since people made it.
Yes. So now there are facilities around the country that maintain these animals germ-free.
Wow.
And anytime you want to you can rederive any animal germ-free, and you just have to take it through this process.
Mm-hmm.
But now you can use the germ-free moms that are currently in the bubbles as surrogate mothers.
Yeah, yeah. And I don't necessarily know if this is your area, and I don't think we'll go into this too much, but I want to ask because it's interesting. So you said, when these mice were created, a C-section is done, the little developing mice were taken out. One thing that's kind of implied there is that the microbiome they would have had would have come from the birth process. It would have been seeded by the mother to a large extent. Can you talk a little bit about that? Is that basically how the microbiome of most animals gets started?
Yeah. So at least in mammals, that is the case. You're fairly sterile when you are born.
Mm-hmm.
There's contention as to whether there's microbes in the placenta and things like that. But you're basically sterile when you're born, and you acquire your first seeding is really through the birthing process. So through the mom's vaginal canal is where you're really seeded with your microbiome. Now, if C-section babies often have a very different microbiome than a baby that's born via a vaginal birth. And then as you grow up, you acquire microbes again from your mother. You'll get some from your father and siblings, and then your environment. But really that first seeding is from the initial birthing process.
And do we know it sounds like there's quite a big difference between babies born naturally, passing through the vaginal canal versus those born via C-section. I was a C-section baby myself. Do we know anything about humans, how those two populations differ?
Yeah, there's a lot of epidemiological studies done on this. Um, Babies that are born via C-section tend to develop more autoimmune disease later.
Interesting. I have an autoimmune condition. Yeah,
I'm sorry to hear that.
it's minor.
And maybe later we can talk about some of the stuff we're doing with early life development.
Yeah, yeah.
Um, but that is one of the major things that is different with babies that are born via C-section
Mm-hmm.
and via vaginal birth is that
Mm-hmm.
they develop things like IBD.
Yeah.
Another disease that the tendency is that if you're born via C-section, you have a higher chance of developing that later. So,
Mm-hmm. And I guess if we sort of put that together with what you said about the germ-free mice, if germ-free mice don't develop a very effective immune system because they completely or largely lack a microbiome, I think, if you compare the C-section babies to the natural birth babies, it would imply at the very least that their immune systems get trained up in a different way. And perhaps this is related why you see these things with autoimmunity.
yeah, that's right. I think, I said those microbes are really important in training your immune system to fend off a pathogen, but the microbes are also equally important in teaching your immune system when to not respond to things, including
Mm.
like your own tissues.
Yeah,
Like
yeah,
really what autoimmune disease is when your immune system is inappropriately recognizing your own tissues.
yeah, yeah, yeah.
So
And so I
microbes
would,
are important in that.
yeah, if I had to guess, I would guess that a natural birth results in a microbiome that's probably more dense and diverse in composition.
Um, yes and no. I don't know if the studies are clear on that.
Mm-hmm.
I think it's that the microbes are very different.
Just
So when
different.
you're,
Okay.
yeah, when you're born via, a vaginal birth, there's a lot of lactobacillus that you're introduced to. There's a lot of, Candida that you're introduced to. Whereas if you're born via a cesarean birth, um, then you tend to have more skin microbes.
Mm-hmm.
So I think you're not
Yeah.
supposed to be exposed to those like
Yeah.
right at the moment yet.
Yeah. I've even heard that babies born by a C-section tend to have microbiomes that start to resemble the skin microbiome of the doctor, whoever delivers the baby.
That's interesting. I've actually never heard that, but that's funny.
I
It
don't.
doesn't surprise me.
I have not verified that, but I think I read that somewhere. Okay, so like the composition of the microbiome obviously is very important for how the immune system gets trained up. It's natural to think how that would be related to things like autoimmune disorders or just your general ability to fight off pathogens. So the other thing I want to ask you for some background here is, one of the things that seems like it's a fairly rock solid part of the literature that I hear over and over again is when we talk about nutrients that are good for your microbiome or that promote a healthy and diverse microbiome, the one that comes up the most is fiber. And you mentioned that fiber, things like cellulose, we don't digest them, but as people often say, they're food for the microbiome.
Mhm.
What I want to unpack that a little bit more. What do these microbes do with the fiber, and what are they producing that then turns out to be good for us?
Mhm. Sure. Um, so this is not, I will disclose that this is not my area of great expertise. We don't study this a ton, but of course, it's one of the bigger things in the microbiome literature is diet. So the fiber that there's lots of different types of fiber.
Mm.
So first, I'll say that not all fiber is created equally, and not all fiber is going to be metabolized by the microbes equally, and not all fiber is going to have a benefit. So
Mm.
there's been a couple studies that I've seen where the fiber inulin, which is one of the ones that a lot of people will take, actually is really bad for certain diseases. It's good for some, but bad for others,
Hmm.
based off of, how the microbes metabolize it and how it influences your immune system. But I think, one of the best metabolites that's studied because of fiber production is short chain fatty acids.
Yeah.
I don't know if you've heard of these before, but everyone studies short chain fatty acids.
Mm-hmm.
And these just seem to be globally good for you in that they induce an anti-inflammatory response.
Hmm.
They can also act as fuel for your intestinal epithelial cells. So your intestine is lined with these epithelium. That's kind of one of the major cell types that
Mm.
creates a barrier between the microbiome and the host.
Hmm.
And these short chain fatty acids are solely made by the microbiome, and they can be fuel for those intestinal epithelial cells and keep them, keep that barrier tight.
Yeah, yeah. Well, that's interesting. So these short chain fatty acids, maybe we'll talk about chain length later, but shorter, sort of smaller fatty acids, they can be used directly as fuel. It sounds like, so if you've got microbes in the intestine, they can take that fiber, make these short chain fatty acids. It seems like it's a really good, efficient way to locally have energy for those cells. They probably don't need to spend as much of their their time and energy making fuel if you can have these microbes producing it locally right there.
That's right. Yeah.
And so what would be the difference between just at a high level? So when we talk about a short chain fatty acid being used for energy as opposed to something like glucose, is there anything worth mentioning there? Is like one of them better or preferred by our cells as a fuel source?
Glucose always seems to be the preferred fuel source for everybody, our cells and the microbes. I don't think that I can intelligently comment on whether or not short chain fatty acids or glucose are preferred. I think it all involves the environment, what the cell is doing, what the cell needs.
And I guess one last question I have here is, so they produce some of these microbes produce these short chain fatty acids, which can be used by cells for energy and perhaps for other things. Are there other ways to get short chain fatty acids, or is this the only way?
So our body doesn't produce them. It's, the fermentation byproduct of fiber that produces them. The other way to get them is people do synthesize these, like chemical companies synthesize them. So for instance, in the lab, we can buy, because there's lots of different short chain fatty acids. There's butyrate, there's propionate, and you can purchase these and you can orally take them. And a lot of people will do that in their mouse studies to show that, this short chain fatty acid is sufficient to have this effect.
But other than that, other than these products, is this not really a component of natural sort of whole food diet?
Not that I'm aware of. No.
So I know that there's, the microbiome's been connected to, all sorts of stuff. And we'll talk about some of that. Obviously it's connected to, gut issues that we can talk about, inflammatory bowel disease and things like this. But it's also been connected to obesity. And, we're going to talk about one of your papers that you shared with me that's really, really interesting here. But before we get to that in particular, what's sort of known, what's out there in the literature right now in terms of the connection between the gut microbiome and obesity?
So, what is currently known is that I guess on a very basic level, is that the microbiome, when it is less diverse, if you will, is associated with increased obesity. So what does diversity mean? We have these kind of like arguments in the lab about this. I think just on a very superficial level, if you are missing certain microbes, and we are still uncovering what these specific microbes are and what they do, you seem to be more prone to gaining weight. So your microbiome composition seems to be very important in how you gain or lose weight.
And in principle, like what would be the ways we imagine that could happen? So off the top of my head, I could imagine, well, maybe certain microbes are making certain key nutrients or producing metabolites that have to do with energy expenditure in our bodies. Maybe they're regulating how much of the calories we're actually absorbing from the food that we put into our mouths. What's sort of the space of possibility there?
Yeah. So I think one of the first mechanisms ever discovered was really from Jeff Gordon's group. And this was when they basically took a microbiome from a obese mouse and transplanted it into a lean mouse, and that animal gained weight. And what they showed is that microbiome was really good at extracting energy. So it was able to extract more energy from the food, and then it gave more energy to the host.
I see. Did those mice actually eat more, or was all of it accounted for just by absorbing more?
In the transplant, the animals did not eat more. It was more about them just having more nutrients and
Hmm.
extracting more energy. The obese animal used for the microbiome transplant, it was a leptin deficient animal, so it just
Okay.
constantly eats. So,
Constantly eats.
there's no doubt there's a component of, diet in influencing the composition of your
Mhm.
microbiome.
But just that microbiome change was accounting for a lot here.
That's right.
Yeah.
hmm
Interesting. So tell us a little bit about this paper that you shared with me. Can you set that up for us in terms of what kind of questions you guys were asking and what the basic setup is?
Sure. So this was one of the first studies I embarked on when I started my own lab. So during my postdoc, I had been studying a bacteria that we had discovered was able to prevent inflammatory bowel disease in mice. And the mechanism by which it was doing that was inducing a T regulatory population, which is an anti-inflammatory cell. And that's how it was able to suppress inflammatory bowel disease. And we identified a pathway within the cell that it was able to induce this Treg population. So it was basically inducing it by making a polysaccharide that bound to a toll-like receptor on a T cell. So and a toll-like receptor is one of these receptors in innate immune cells that's able to recognize many components of bacteria. So they're
Mhm.
somewhat promiscuous.
So these receptors are for they're for sort of recognizing what is and is not a pathogen or a human cell.
They are, but they're broad in scope. So they
Mm-hmm.
recognize pieces of bacterial cell walls or bacterial DNA,
Mm-hmm.
and they can do that whether it's a commensal or a pathogen. So they're not really great at differentiating between the two.
I see. I see. They can just sort of see like this is our cell, this is human cell or not.
Yeah, basically, yeah. So people are still trying to understand how these cells help distinguish between commensal and pathogen.
Mm-hmm.
But it made us really interested in that pathway with respect to, because it seemed like this pathway was really important in inducing a healthy homeostatic intestine. So we made an animal. So I should preface this, like I did not start my lab thinking I was going to study obesity. I
Mm.
would I thought we were going to, find a pathway that was involved in IBD. But you just follow the science. So we made this animal where we got rid of a signaling molecule that's downstream of toll-like receptors. It's called MYD88, and basically this is a signaling molecule downstream of most of toll-like receptors.
Mm-hmm.
So by getting rid of it, you're essentially disrupting the ability of whatever cell type. We were looking at a T cell at the time,
Mm-hmm.
but you're disrupting the ability of that T cell to signal through any of those toll-like
Mm-hmm.
receptors.
So does that mean basically the immune system can recognize when it meets a non-human cell through this toll-like receptor, but it can't act on that because you're sort of blocking the information flow?
That's right, exactly. And I should say that we only knocked it out in the T cell. So the rest of the cells in the mouse's body were able to make that
Mm-hmm.
recognition. So they were still active. So when we made that mouse, one of the major phenotypes of that mouse is that it had a significant reduction in the amount of IgA it produced in the gut.
Hmm. And you said we produce normally a lot of it.
A ton of it, right? But this mouse, when we knocked out this pathway, was not making as much.
Mm-hmm.
And then if you let those animals grow up to old age, most people do not let mice sit around in their facilities beyond 8 weeks because it's
Mm-hmm.
very expensive to keep mice. For some reason, my graduate student decided she was going to not cull any mice and she was going to let them grow up to be like over a year old.
Mm-hmm.
And these animals developed this spontaneous obesity. They were like really fat. They were like, she always called them like little pancakes.
Hmm.
Um,
But late in life, did it develop this late in life?
Late in life, yes.
Huh.
It was not immediately, we didn't observe it in like an 8-week-old animal.
I see. So as the mice are growing up and going through adolescence and things and becoming young adults, they looked normal macroscopically to the eye.
That is correct. Yes. So they did have this IgA deficiency
Mm-hmm.
at that early stage, but it wasn't until later in life that they developed the obesity. If you did put the animals on high-fat diet, they developed the obesity quicker.
Mm-hmm.
So you could start to observe it. But yeah, it seemed to be a spontaneous phenotype, which is very rare to see in animals, actually. Normally, you have to push the obesity by giving them a lot of fat or changing their diet in some way. So this was a really striking phenotype to us because we had knocked out this immune pathway. We thought it was going to be involved in gut homeostasis, but these animals were getting fat. So it made us think about the microbiota. I'm a microbiota lab, so of course we thought about that. So we did transplant studies where we took the microbiota that formed in these obese mice and we transplanted them into germ-free wild-type mice that did not have this knockout. And those animals got fatter.
With the same timing, with the same developmental timing?
No, they actually gained weight faster than these other animals.
Okay.
So what I will say is when you take that microbiota from the knockout animal and put it into a wild type animal, over time, the immune system in that wild type animal will kind of reshape that microbiota to be more of a healthy microbiota, if that makes sense.
Yep. And so in the genetically engineered mice that develop obesity in late age, did you characterize sort of how that's developing in terms of, are they expending less energy? Are they eating more? Or does it appear to be one of these things where they're just absorbing more from what they do eat?
Yeah, so we did all of that characterization and the animals didn't eat more. So they were eating the same as the wild type animals. They were not expending more or less energy than wild type animals. The major difference seemed to be that they were not absorbing lipids as well. So there were more
not absorbing them as well.
Not absorbing them as well. Yes. So there was more left in the stool that would be excreted. Oh, I'm sorry. The knockouts were absorbing more. The wild type.
Okay. I was going to say.
I said that backwards. So that was.
So yeah. So basically, they're pulling more fat out of the food and absorbing it into the body.
Yes.
Okay. And is that so? A couple questions here. So. One, is it a specific type of fat? Are they absorbing saturated fatty acids, or is it just sort of everything?
So the ones that we really noticed a difference in were these long chain fatty acids. I mean, these are just like basic, like once your lipases break down the fats that you eat, you get these long chain fatty acids. They bind to specific receptors on intestinal epithelial cells, and then they're taken up into the intestinal epithelium and then basically packaged into these chylomicrons and sent throughout the rest of the body for use. So those were the long chain fatty acids were the ones that we were noting were really different in our animal.
And is there, I'm asking from a place of ignorance here. So chain length is important for a variety of things when we start thinking about the different fats that animals consume and what they can be used for. So you're talking about long chain fatty acids here. These are the ones that are being absorbed more by these genetically altered mice. Earlier, when we were talking about fiber in the gut microbiome, they were making short chain fatty acids. Is there any differences we should talk about here in terms of the short versus the long chain fatty acids that you think are relevant?
Other than their chain length, I mean, the long chain fatty acids I'm talking about are ones that come from the diet that have been acted on by the lipases that are excreted from your pancreas. So these are more dietary fats, and then our host enzymes make them into these long chain fatty acids. The other ones, that we were talking about before are products of the microbes. Yeah. But at the end of the day, molecularly, it's a difference in chain length. So I don't know that there's anything to speak of beyond that.
Yeah. But I guess, one of the questions, so it's I don't know this for a fact, but this would be what I guess. So the short chain fatty acids are more readily burned as fuel. That kind of makes sense to me that the shorter ones would be used as fuel more. And then the longer ones, are they just more apt to be used for energy storage? And then maybe this is, yeah.
I see where you're going with this. Yeah. So short chain fatty acids can be absorbed more readily by an intestinal epithelial cell and used right then and there.
Yeah.
Long chain fatty acids tend to bind to receptors on the surface of the intestinal epithelial cell and then taken up and then delivered to the rest of the body. So I guess what you could say is short chain fatty acids are maybe used more locally, even though they are delivered throughout the body as well. Whereas
Mhm.
the long chain fatty acids are packaged up and then sent to
Mhm.
the rest of the body. So
These long chain fatty acids, they're not just a repository for energy. They can act as signaling molecules. It sounds like, and
that's right.
and they just need to sort of be processed more before they can be used.
Yes, that is correct.
Okay. So what comes next? You've got these genetically engineered mice. They get fat in late age. They've got this interesting immune phenotype that you've engineered into them. What comes next here?
So the next question to us is, well, I guess there's two, but here we know it's a microbiota driven obesity.
Mhm.
So one of our initial questions was what microbes are driving this obese phenotype? So I think normally when you do these experiments, you're looking at the obese mouse and
Mhm.
you're asking questions about, well, what microbe is there that's making them obese? Because that's generally like what pathogenesis people think about, like what microbes there causing a disease. And we did do those studies. We looked, and there was nothing that really jumped out at
Hmm.
us too much. The one thing we really noticed was different when we knock between the knockout and the wild type animal is that the knockout animal seemed to be missing a group of organisms within the Clostridia.
Hmm.
That was the biggest difference in the microbiota that we noted is really microbes missing in the knockout.
I see. So these animals don't have certain Clostridia bacteria.
That's correct.
And what do we know about those bacteria? Like, what's our baseline knowledge there? Are they doing something in particular that's important?
So at that point, we knew that some members of Clostridia could induce Tregs. That had been shown in the literature. But beyond
Hmm.
that, we didn't know a ton.
Hmm.
Because I should say this is like a huge class of microbes,
Okay. Okay.
and they're highly uncharacterized.
I see. So there's all sorts of different species that are in this general group.
That's right. And when we had discovered it was this group, when was that? 2019, we published that paper. So much more has been done regarding sequencing and taxonomy of this group. A lot of things have changed.
Hmm.
But what's unique about these bacteria is that most of them are spore forming. So a spore, a bacteria that can undergo sporulation, it's a way of protecting itself. So if it undergoes environmental stress, it can basically go into its spore form and it can resist things like antibiotics. It can resist desiccation, um,
Hmm.
it was a nice way for us to be able to isolate these organisms because we were able to take a healthy microbiota and extract all the spore forming organisms, which were mostly Clostridia, and we could give them back to those obese mice and we can make them lean again.
Hmm.
So that was kind of the proof of principle experiment that we did to show like these are the organisms that are making these mice lean, helping them to stay lean.
I see. So you've got this genetically engineered mouse. It's missing this key component in T cells of the immune system.
Mhm.
Um, and they get fat in late age and they seem to be missing certain bacteria, but then you can give them the bacteria and it reverses the obesity phenotype.
That's correct.
Hmm.
Yeah.
How do you give them the bacteria, and how is it a full reversal? How quickly does this happen? How dramatic is that reversal?
That's a good question. So we give the bacteria via an oral gavage. So it would be the equivalent of a person like just taking a pill.
Yeah, yeah, it's like taking probiotics.
That's right. Yes. And we give it to the mice every other day, so it's not a ton. The reversal is pretty dramatic. And actually, I shouldn't say the reversal. It is the prevention because we basically start giving these Clostridia to these animals before they develop
Ah, I see.
obesity
Okay.
because we knew that they would develop obesity later in life.
Yeah.
So we gave them the organism early in life.
Hmm. Hmm.
So we were asking if we could prevent the onset of obesity if we provided these organisms. So that's what we did. We still have to do the reversal experiments. We haven't done those.
Yeah, yeah. Interesting. Okay, so genetically engineered mice, they're defective in their immune system in this particular way. They
Mhm.
don't have certain microbes that they would otherwise have. They develop obesity late in life, unless you give them these bacteria earlier in life, and then they don't develop obesity.
That's right. You're really good at this, Nick.
Thank you. I'm just trying to make sure we're following. So, so how do you interpret that so far? Is it just, and I may have missed this. Were you giving them back just one species, or is it sort of like a bunch of different species in this family of bacteria?
Yeah, it was a big group of
Big group.
bacteria.
Okay.
It was probably 40 to 50 different types of organisms, all of them
Hmm.
being spore formers, but all of
Hmm.
them being part of Clostridia.
Hmm. And then I assume you characterize sort of what was going on under the hood. The lipid absorption probably then just looks normal in these mice.
That's right. Yeah. When you put in those Clostridia, you resolve the increased absorption of those lipids.
Hmm. And so how do you, so, one thing people might think about here that I think immediately comes to mind is, so many people have had experiences or know of people who've had experiences where, even sometimes within the same family, or, your friends or whatever, you seem to be eating more or less the same diet. It's not obvious that one person's like completely gorging, more than the other person,
Yeah.
or even people that are, really trying to lose weight and they're cutting back and they fail to do so. One person's lean, one person's not lean or obese, but they seem to
Mhm.
be eating comparable diets. This, starts to kind of sound like that, where you could eat the, it's possible. This is, I mean, I think what this shows is it's possible for two animals to eat the exact same diet and have dramatically different results in terms of body fat deposition because the microbiome is regulating how much is being absorbed.
That's right. That is exactly right. And I think it's still not a very popular thing to say because
Mm-hmm.
most people think it's, energy in, energy out. What you eat, if you expend the energy, you're going to be thinner. But what you just said is right. We just showed two animals they ate the exact same. They did the same movement, but one got obese and one didn't. And it was
Mm-hmm.
all based on the types of microbes they have.
Hmm.
So to me, this this is a really important finding because this says the types of microbes you have are going to dictate your body composition, and this is going to dictate, health.
Mm-hmm. And one thing that's interesting, unfortunate, you might say, a little tricky, is um, these mice don't develop obesity till late in life.
Mhm.
So it's not like you could see something happening earlier on. Um, how do you start to think about like what that might mean for humans and sort of detecting it's sort of like they go through their entire life and something is different about them under the hood, but they don't develop
Yeah,
this phenotype till later in life. How do you start to think about that from more of a human perspective?
sure. So I think there's probably two things to say there. The first being um, is that we were able to induce these animals to become obese earlier in life
Mm,
if we pushed it with a high fat diet.
I see. So they more readily develop obesity. Is
That's
you
right.
almost might say that's the phenotype, and it just if you don't push it with a high fat diet, it takes a long time to set
It takes
in.
longer.
I see. I
Right.
see.
So diet was a really important component here.
Hmm.
And we can talk more about why mechanistically I think that is the case. Um, the second thing to say, I just forgot. So maybe it'll come to me in a minute.
Yeah, yeah, we'll come back to it. I
Yeah.
wanted to ask you, so when you look under the hood and you compare the mice you guys made, the genetically engineered mice that are predisposed to obesity, basically, what else, if anything, is sort of wrong with them? Do they have fatty liver disease? Are they insulin resistant? What does sort of the physiological or metabolic health profile look like?
Great question. So it's not just obesity.
Hmm.
These animals develop fatty liver. They don't clear glucose as well. So they're starting to look like they're getting diabetes.
Hmm. So it's basically a
Um,
metabolic syndrome.
it really is a metabolic syndrome, and I
Yeah,
think we
yeah.
use that terminology in our paper a lot. And this is, something that's becoming a huge problem in Western civilization.
Hmm. And the other thing that was interesting is I think you have a result here where you took these genetically engineered mice that develop obesity. And as you said, they're missing certain populations of bacteria in the gut. They're developing metabolic syndrome, and then you just you co-housed them with other mice. What happened there?
Um, so those are interesting experiments. So this is very similar to a microbiota transfer
Mm-hmm.
study. So when you put mice into the same cage, they're coprophagic, so they essentially eat each other's poop. So that's, essentially that's what a microbiota transfer is you are feeding
Yeah.
mice.
So by their nature, mice will automatically do a microbiome transfer.
That's right. So
Yeah.
generally, if you put mice into the same cage, their microbiome starts to converge and look more like one another. It's
I
not
see.
always the case, but for the most part, that happens.
Hmm.
So when you put knockout and wild type mice together, what we saw was actually the knockout animals that were co-housed with wild types. Those animals got, oh my gosh, they got fatter. So they took on the phenotype of whoever they were, those mice that they were caged with.
Hmm.
Um, those experiments are really nice because you can start to follow what microbes are moving to what animal. And that was really one of the key experiments that helped us identify this Clostridia population.
Hmm. And so when you give these animals high fat diets, can you say a little bit? So what does high fat mean exactly in terms of total fat? And is it important at all what the specific sort of fatty acid composition or the other composition of the food is?
So our high fat diet is a 45% high fat diet, which is actually low for what other people do in the field. A lot of people give 60%
Hmm.
high fat diet. Most of the fat is coming from a palmitate in these diets, although not exclusively. And I think the source probably does matter, but that's
Hmm.
just the diet that we.
Is that a saturated fatty acid or unsaturated?
I don't know. I think it's saturated.
Okay. I I'm
You,
I'm I'm
you
with you.
quote
I'm
me on
like
that.
80% sure it is. But yeah. So it's 45% fat. I don't know the numbers off the top of my head, but I believe like what would be a typical average American or someone eating the so-called Western diet? I think it's, roughly 50-60% carbs and maybe 35-40% fat. So, I guess kind of generally in this ballpark.
Yeah, I think that's one of the reasons we used the 45% high fat diet because we felt like it was more akin to what a human might
Hmm.
consume as opposed to
Hmm.
60%, which just seemed outrageous.
Yeah,
But
yeah.
I guess if you're eating McDonald's all the time, you might hit that.
Hmm. And so you co-house these mice. You've got the obese one and the non-obese mice. The lean mice become obese. So do their phenotypes converge? Do the obese mice become a little bit more lean, and the lean ones become a little bit more obese, or do the lean mice really just take on the obese phenotype?
Um, the lean mice take on the obese phenotype.
Hmm.
It was not a convergence, which I thought it would be.
Hmm.
Um, yes.
Yeah. So, well, yeah. What do you
Sorry.
like? Do you have any thoughts on like why that might be, or is that mysterious at this point?
Yeah. Um, so I think what was happening, and again, what I said before is that these experiments were one of the critical ones to tell us that, like, I help us identify who the important microbes were.
Hmm.
So, because what you do is you sequence prior to the co-housing experiment,
Hmm.
and then you sequence after the co-housing experiment.
Hmm.
And what this does is it really helps you to, because the microbiota is incredibly complex. So
Yeah.
we're always asking questions about, who is it?
Hmm.
So when you do these sequencing experiments, then you can identify the key microbes. So what we did is we could find the microbes before and after transplant, and you could see the Clostridia, you could see those Clostridia moving around. And
Hmm.
wherever the Clostridia were, the mice were lean.
Hmm.
And wherever they were missing, the mice were fat.
Hmm.
So that's what really highlighted um, who was important here.
Yeah, and it's kind of a convenient or like optimistic type of result in the sense that okay, you're showing that you can basically prevent a phenotype by just introducing the right bacteria. You can do this orally. Obviously, people not just in the scientific world, but out in the rest of the world, probiotics are a big thing now. There's lots of marketing around these things, but the implication is that something like this could work in principle if you take an oral pill that contains the spores of the right bacteria, at least early enough in life, that can, at least in these mice, be sufficient to prevent the onset of something like obesity.
That's right. And so we have a study that's in review right now because I told you that these Clostridia were like 40 to 50 different organisms. That's
Mm-hmm.
really hard to grow that up, put it in a pill.
Yeah, yeah.
So we've been really interested in identifying like who is the member of this consortia or members who are really driving the lean phenotype.
Hmm.
Because just as you said, it seems like a really great way to just stay lean is to just make sure that you have the right bacteria.
Hmm.
And we've identified one particular species in that group that's driving most of the lean phenotype.
Hmm.
And we can purify that organism out and give it to mice and make them lean.
Hmm.
So this is really exciting to me because now we can also go and look at the microbiomes of humans, because there's a ton of studies that characterize the microbiomes in humans.
Hmm.
And if you look at humans who are obese or have type 2 diabetes, this particular species is missing.
I see.
So it really makes us feel like, what we're finding in mice might be applicable to humans.
I see. Yeah, I was actually going to ask that next. I think is how much does the microbiome of mice match humans? But it sounds like, at least for this species, like this is we both have this in our microbiome. We can both be missing it, and there's at least a correlation between not having it and being obese in humans.
That's correct. Right.
And so, is that like coming up next, or is someone going to test this in humans?
We are trying, yes. It's a little bit trickier to do these types of things in humans because you have to grow up the bacteria to a certain like cleanliness level.
Right. And I think one of the tricky things here is, well, so if we go back to the question of reversing the phenotype versus preventing the phenotype from developing, you were able to prevent it from developing by giving mice the right bacteria, throughout their life. The question is, can you, if you do the experiments where you reverse it in adulthood, that would imply that maybe something like that could work in humans. If that didn't work in the mice, then you've got a greater challenge, because you've got to somehow detect that people are deficient in these bacteria early enough in life, and then supplement them with it before they ever develop obesity. How are you sort of thinking about all that?
Yeah, and this goes to the thing that I forgot to say earlier, because you asked me, like, how do you parse out people who might be more prone to developing obesity or not?
Yeah.
I think if we can identify the specific bacteria that are really driving the phenotype, kind of like what we're doing now, it's really easy to detect these bacteria, various molecular methods, PCR, for instance, in people's stool. And one could imagine you eventually will get to the place where people are sequencing their microbiomes, let's say, when they're younger, and asking, like, am I missing this microbe that will make me lean? And if so, then you could say, I'm going to supplement myself so that I can prevent late onset obesity.
Yeah.
So I do think that as we progress on in this field, that you'll be able to make these types of predictions by looking at the microbiome early in life to see what you have and
yeah. So I guess technologically, it's now feasible that at some point, if humans organize themselves, our healthcare system in the right way, you go in for your yearly checkup. Maybe you get some blood work done to look at your lipids and things like this. This tells you, about your cardiovascular disease risk and other things. But maybe you also get your microbiome checked, and we can see, okay, the 6-year-old actually is missing the Clostridia or whatever species that we think should be there, and then you can supplement accordingly.
Yes, I think that this is like one of the futures in clinical practice, is that we're going to, a lot of people are sequencing their genome to see what genes might predispose them to different diseases. I think eventually we're going to be doing the same with our microbiome. We're just still at the stage of trying to figure out what bug's doing what.
Yeah. And so in your experiments, obviously, you've got all of these tools and these experimental conditions that you guys can engineer in mice. And this allows you to, start figuring out mechanisms and doing all the work we just talked about. When you think about sort of naturalistic obesity, let's call it, in humans, we're not engineering humans to miss, a component of toll receptor signaling. We're not doing like things like that. And yet obesity is what it is. It's spreading like wildfire, basically. What are and let's just suppose that, like in certain mice, at least some of this in some people has to do with microbiome deficiencies. What are the plausible sources of those deficiencies in humans? Is it the way that humans are born? A lot of people born from C-section versus vaginal birth. Is it the use of antibiotics in early age? How do you start to think about what could be disrupting, what's plausibly disrupting the microbiome in humans?
I think the two easiest answers to that question are going to be diet and then um, antibiotic use early in life. So some of Marty Blazer's work has shown that if you treat animals with antibiotics early in life, you can even remove those antibiotics and let them grow up.
Mm-hmm.
The animals that saw antibiotics more readily got obese.
So merely giving animals, mice in this case, antibiotics early in life, taking it away, everything else is normal, they're more prone to obesity.
That's right. And that could be because what that suggests is because you're deleting some specific bacteria.
Mm-hmm.
Diet is also another major one. I don't think we fully understand how diet regulates the composition of our microbiota, but going back to this bacteria that we're studying that prevents obesity, we have found that if you give the animals a high-fat diet, this particular microbe actually cannot colonize the gut anymore. And then if you try to grow it up, if you take it into the lab and grow it up in a high-fat diet media, that animal, or I'm sorry, that microbe can no longer grow. I think microbes the fats can be toxic to some microbes.
I see. Yeah. And that makes sense in principle. Like, as we mentioned, these fatty acids, fatty acids aren't just a source of potential energy. They're signaling molecules. They
Right.
have information they can stimulate in cells. So basically what you're saying is the idea here would be if you happen to, obliterate the microbiome or lose a species through antibiotic use or whatever, if you then if that person, if you sort of let that person or let that animal develop, if they have, say, a high fat diet, that could actually prevent the colonization of the bacteria that you want. But if you don't have a high fat diet, perhaps that wouldn't. And so basically the dietary context can dictate whether or not, a certain bacteria species can grow back. And then that's going to have implications for things like obesity in this case.
Yes, that's right. It's very complex, all of it.
Yeah. And, it's like one of those chicken and egg things when we think about like what's happening in the naturalistic case. So obviously changing your diet can change your microbiome. Your microbiome can change how your diet is used by your body. So like how readily, like, do you think it's feasible, or do we know, for example, that, let's say someone is eating a high fat diet and they have for most of their lives. If you change the diet, in a fairly dramatic way, is that a reliable way to change the microbiome, or can the microbiome sort of get locked in a way that's really hard to change?
Well, I certainly think that if you're on a particular diet for a long time and you're not allowing colonization of those organisms, they're not going to magically come back. So I think you would have to do something along the lines of changing your diet and then reintroducing the microbes that you've lost. You would have to do both for sure, because I do think, as you said, the microbiome can get locked in, so to speak, because only so many colonize.
When you think about, like, when I start to think about different strategies for doing this, I start to think about things like, okay, so when you say introduce new bacteria, there's a question of if we're talking about humans, it's like, do you take the probiotic pill that has been made to have these bacteria, or do you say go on a fasting regimen and then eat a diet that will be conducive to that bacteria blooming? How do you think about that? Do you think a healthy microbiome can often come as a natural consequence of eating a healthy diet, whatever exactly that means? Or are we going to need to really like put bacteria spores in a pill and do it that way?
I guess I envision that you can get to the place where you're just missing certain microbes. And then unless you reintroduce them to the system via a pill or a fecal microbiota transplant, just eating a fiber-rich diet is not going to get you back to the place you need to be, because those organisms are gone. And once they're gone, unless you reintroduce them to the system.
If the origin of these Clostridia bacteria in this case is not dietary,
Right.
Like, let's say, like, I don't know, do they come from like the vaginal microbiome? And after you're born, you're only born once, so you don't get a second chance to see your microbiome. So maybe you need to introduce them explicitly. Do we actually know what the origin of these bacteria were that you were studying?
So these organisms aren't necessarily from the vaginal tract
Hmm.
of mom. These probably get seeded later. I mean, again, this field is still in flux. We're still
Yeah, yeah.
learning where you get all these organisms, but you really only find them in the intestine of humans.
Hmm.
So it's not something that's getting introduced by food. It's got
Hmm.
to be getting introduced via your interactions with your family or humans.
Hmm. Hmm.
Um, but yeah, you don't find them. They're not being introduced during the birthing process.
Yeah, yeah. So, obviously we talked about germ-free mice and giving mice antibiotics early in life that can predispose them towards obesity. How do
Mhm.
you, just as a human, not necessarily as a scientist, I'm sure at some point you're going to have to take antibiotics. I'm sure if you've got kids, they either have taken antibiotics when they got really sick, if they really needed it, or they might have to in the future. If and when those things happen in your life, how do you think about managing your life subsequent to the antibiotic use, given everything you know about the microbiome?
Yeah, it's a question I get quite a bit. Right now, most of, if not all of the probiotics that are available for people to purchase are a very narrow spectrum of our
Hmm.
microbiome. They
Hmm.
are lactobacillus and bifidobacteria. These are like the organisms you get in your yogurt.
Hmm.
Um, it's kind of shocking, really, that the probiotic industry hasn't expanded beyond those organisms. But those
Hmm.
organisms in an adult gut are very minimal um, type of microbe in your gut.
Hmm. So they're probably just easy to make and manufacture.
Right.
Yeah.
They are, and they can withstand oxygen.
Hmm.
Some of these other microbes, you're going to have to grow them up in a very strict anaerobic environment. And I
Hmm.
think they're just going to be harder to manufacture, which is probably why we don't see them on the market yet.
Hmm.
Um, so I don't think right now, if we have the right tools to restore our microbiome the way that I would like to restore the microbiome after,
Hmm.
let's say, an antibiotic exposure.
Hmm.
But with that said, for instance, you know, one thing that they're doing is kids who are born via cesarean section, they're now swabbing the mom's vaginal tract and colonizing the baby with that.
Hmm.
That's not something that was done, when I was born, probably when you were born. This is something that's fairly new. So
Yeah.
people are starting to think about more natural ways of replacing the microbiota if we have to do some of these things. Like, some babies just have to be born via cesarean
Right.
section.
Right. Yeah. Yeah.
Um, High fiber diets, right? This
Yeah.
is
Yeah.
what I eat a lot of fruits and vegetables to ensure that I'm feeding my microbiome correctly.
Hmm. And I'm not sure if this is your area, but I do want to go back to some things you said at the beginning. So you mentioned, we talked about fiber as food for the microbiome and the microbiome producing short chain fatty acids. Two things I want to cover. So one is on the input side there. What fiber you're eating. You mentioned there's different types of fiber. Can you give us a little bit more of a sense for the different types and which ones are the fuel for short chain fatty acid production?
Yeah, I don't think I'm going to do that question very good justice.
Okay.
Um, Only because that's not my area of expertise.
Hmm.
If you ever
But
want to talk about that, you should invite Eric Martins onto your podcast.
okay, that's a good tip. I'll look him up. But I guess sort of high level, there's different types of fiber. Not all fiber is created equal.
That is correct. Yes, because I
So
think
just because something says fiber on the nutrition fact doesn't mean it's going to be the one you might think it is.
that is absolutely true. Yes. Like I said, inulin is one that's well studied in the field. It's one that a lot of people can buy off the shelf.
Hmm.
But there have been several studies, at least in mice, that if you give it to mice, those animals will be more inflammatory in their gut under certain circumstances. So I
Hmm.
think this field just needs, certainly more research.
Hmm. So what are some of the other types of experiments that you guys have been working on in the lab? Is it mostly focused on microbiome and obesity, or do you do other stuff as well? It sort of sounded like this was kind of a serendipitous direction you went in.
Yeah, we work in a lot of areas because people at the University of Utah are just incredibly collaborative. So we have
Hmm.
projects in cancer, We have projects in pancreatic development. We have projects in autism. One thing that might be of interest and pertaining to our conversation is the pancreatic development stuff. And this is because it involves an early life perturbation to the microbiota. So I can talk about that one because I think it might be of interest to you.
Yeah.
So I had a really wonderful postdoc in the lab. Her name was Jennifer Hill. She's now started her own lab. And she was interested in understanding how the microbiome could influence beta cell development. So beta cells are a cell type in your pancreas that make all the insulin in your body. Obviously, very important cell type. And it's also the cell type that gets destroyed in type 1 diabetes. So the immune system attacks these beta cells. What's interesting about beta cells is they don't replenish. Basically, they develop when you're very young. So within the first year of life, they undergo this very large proliferative event that sets the number of beta cells you'll have the rest of your life. Also, as we've talked about, the microbiome during early life, that's when you're the composition's in flux. Basically, after 3 years old, your composition pretty much looks like the composition you'll have for the rest of your life. So she was really interested in this early life event and beta cell development. So what she did is she set up this experiment where she perturbed the microbiota during 10-day intervals of the mice during their first 21 days of life. So she pulsed with antibiotics for the first 10 days, took them off, let them grow up to adulthood. She then did it for, another 10-day interval and then another 10-day interval. Took them off antibiotics, let them grow up. What she found was amazing to me, and it was a little bit scary, is that if she pulsed with antibiotics between postnatal day 12 and postnatal day 21, but not before and not after, those animals had significantly fewer beta cells when they were adults. Also could produce less insulin and could not get their glucose as well. So it's like that small
window.
Yes.
Yeah.
Basically ruined how their beta cells developed for the rest of their life.
And so I guess the natural way to begin thinking about this, at least, is, okay, you give antibiotics at some window early in development. That's obviously going to wipe out a lot of the bacteria. Because the microbiome is involved in training up the immune system, that's going to affect how the immune system gets trained. And because something like type 1 diabetes is the immune system inappropriately killing these beta cells, it's very natural to think that there's a pretty straight line to connect here.
Perhaps, yes. We were solely focused on beta cell development and not necessarily looking at the immune system effects. But we did identify that macrophages, which is an innate immune cell, which seeds the pancreas early in life. The microbiota during this early window was important for instructing those cells to go to the pancreas and enhance beta cell development. So the implications in type 1 diabetes we're still figuring out. We think that we've identified the specific microbe that positively influences this process. And if we put it into an animal prone to developing type 1 diabetes, you can get reduction in type 1 diabetes. But what we don't know is how this microbe is preventing that immune destruction. That's something that we're still trying to figure out. But we think this could be used in enhancing beta cell expansion later on in life in people who do have type 1 diabetes because, again, their beta cells have been destroyed. How are we going to replace them? We think microbe might do that.
Yeah. But I mean, it sort of seems like a general pattern or theme with this and, considering lots of other stuff that, if you perturb the, there's multiple ways to say this or think about this. If you perturb the immune system at different phases of early development, especially either directly by creating genetically engineered mice or something like this, or indirectly via alterations to the microbiome, there are many different ways that things can be disrupted, whether that's beta cell function in the pancreas, or any number of other things we might talk, propensity to obesity, presumably even, development of the nervous system or the cardiovascular system or what have you.
That's right. Yes. You're getting it. You're right.
Yeah. What do you think, as as a scientist in this area, and did you say you have kids?
I do. Yes,
So
three.
yeah, and as a mother and stuff, how do you think about, it's a big, vague question, but we do lots of things in our lives, especially in our early lives, and in the newborns' lives, that are definitely disrupting or nudging the immune system in different ways that we probably don't fully understand.
Hmm.
Do you think that maybe we don't think carefully enough about that as a society? I'll just say it that way.
Yeah, I think we're at a point where my oldest child is 19, my youngest child is 3.
Mm-hmm.
I When I had my first child, I very much was in the mindset of I've got to keep this baby so clean. Like if her pacifier fell on the ground, I was
Mm-hmm.
cleaning it.
Mm-hmm.
I didn't want her to crawl around on the ground. Anytime she had a remotest earache, I was taking her to the doctor, giving her antibiotics. But as I started investigating this area, because I had her very early in my career, I
Mm-hmm.
wasn't even thinking about the microbiome when I had her.
Mm.
But my 3-year-old, I treated her very differently. If her pacifier fell on the ground, I actually just gave it right back to her. I didn't wash it off. I made sure that she was eating like foods high in fiber. She was eating broccoli and grapes, not, I was really thinking about like, I've got to feed her microbes. And if she did have an earache, I would let her struggle with it for a little because a lot of kids can just fight it off. You don't have to
Mm-hmm.
give them antibiotics to clear it
Yeah.
off. Um, so I think the way I raised my first child is more in line with how our society thinks about microbes. Like you got to get rid of them.
Mm-hmm.
Um, but I think we need to let our kids get a little bit dirtier. So as I hope that answered your question.
Yeah, yeah. No, I mean, this ties into something I've talked about with others before, which is I think they call it the hygiene hypothesis. I guess
Yeah.
the broad idea here would be that it's actually our cleanliness, our taking of antibiotics, our hyper, cleaning of pacifiers, and constantly always washing our hands and always, being clean, getting
Mhm.
rid of the microbes. This is actually causing us certain problems, at least some of the time, because it's affecting how our body's interacting with the microbiome and with these pathogens. It's affecting how the immune system develops. Is the hygiene hypothesis something that is plausible or that sort of ties into any of this?
I personally do think that at least a lot of the studies in mice and the epidemiological studies in humans, because again, you can't do causal stuff in humans, they support this idea that not having enough microbes can predispose you to certain diseases. And you said this before, it's certain diseases.
Mm-hmm.
Obviously, there are bad infectious microbes out there, and we don't want to have our kids exposed to those. We don't want to be exposed to those. Um, but I absolutely think that the hygiene hypothesis is plausible. And many people refer to it as the microbiota hypothesis. So maybe it's not just about because the hygiene hypothesis is more about not seeing infectious microbes.
Mm.
And that the whole process of fighting off an infectious microbe and clearing it helps to train your immune system. But some people refer to it as the microbiota hypothesis because thinking more about the commensal microbes and how they're training your immune system. But
Mhm.
yeah, I buy into it.
Yeah. And so in obviously sharing the universal disclaimer with everyone that none of this is ever medical advice, and we are not
Thank you.
physicians. How do you, I want to go back to something you said that I was interesting about, raising your children and, letting them, fight off the infection if they can, at least for some time. How do you like make those determinations around like when to use antibiotics? So the default for a lot of people, as you mentioned, was, and even for me growing up, was okay, the child is sick, give antibiotics immediately, even if it's a viral infection.
Right.
How do you make the assessment of like when it's appropriate to use antibiotics and not, at least for yourself?
So we definitely talk to our physician, and we always go in with a disclaimer that, we don't want to use antibiotics when they're not necessary. And I think more and more clinicians are not prescribing antibiotics as much as they used to. Like, for instance, when I was young,
Mm-hmm.
I remember that pink bubblegum tasting ampicillin. I think
Yep.
they still give it to kids now. I had that so much when I was a child.
Mm.
And when your baby's sick, you just want to make it better. So
Right.
a
Right.
lot of parents go in and they beg their doctors to give them an antibiotic even when it's a viral infection.
Mhm.
But we make the decision based off of a conversation with our physician and then watching, of course, our daughter. We monitor her fever and her pain levels. And
Mhm.
so it's an interaction with our doctor and then us basically seeing how much that child can tolerate before we'll use antibiotics. Obviously, you want to catch it before it gets too bad.
Mhm. Yeah, but you just use, I guess, probably a lot of intuitive feedback. Like, a mild fever is going to be less of a concern than a crazy fever.
Sure. Yes. You're right. Monitoring those numbers. We definitely look at that. You never want to get it too high.
So what is sort of on the horizon for your lab, just in general, but especially related to this obesity phenotype that we talked about today?
Yeah, so related to the obesity phenotype, identification of the specific organism has been really monumental because now we can start to really tease apart the molecular mechanisms by
Mhm.
which this bacteria keeps the animal lean. We've identified one molecule that this microbe makes, which just so happens to be a lipid.
Hmm.
So it's making some sort of lipid that then makes the host lean.
Hmm. Do you know what is it?
It's like a unique bacterial lipid. It's
Huh.
uncharacterized, so we're trying to figure out exactly
Mhm.
what it is. Um, we think that it blocks ceramide synthesis in the gut, which is having a lot of ceramides in the gut is associated with obesity. So we
Hmm.
think that it's making a lipid that blocks this obesity pathway.
Hmm. What are
But
ceramides exactly?
so ceramides are just they're sphingolipids. It's kind of a different type of fat.
Mhm.
Sphingolipids are really important, for instance, in cell membranes. So they're really important and good for you lipids. But if you get too many of these types of sphingolipids, it can change the energy balance in your gut.
Hmm.
So they have been associated with obesity, cardiovascular disease, because if you get too much of them, you can disrupt cell membranes and
Mhm.
cells can start dying. Yes, so the future of our research then is to really identify these microbial molecules, because of course we want to understand the basic mechanisms, but also, a big part of me wants to be able to utilize these therapeutically. So another direction of my lab is trying to test this particular organism and the molecules that it makes in humans. So
Mhm.
we're moving in that direction, which is slow.
Interesting. Um, this has been fascinating, Dr. Round. Anything that you want to reiterate from our conversation or final thoughts you want to leave people with related to microbiome, diet, and obesity related stuff?
Yeah, I think the thing I always end with, and I've probably already said this a million times as we've been conversing, is I really encourage, especially the research in this field, the researchers to start focusing on the microbes that are present during health, identifying those beneficial microbes. I mean, we still have this mindset of finding microbes that are associated with the disease state and always studying kind of the infectious bad organisms.
Mm.
But I think we can really learn a lot by just kind of reversing that thinking.
All right, Professor June Round. Thank you for your time. This was great.
Thank you, Nick, for having me. I appreciate it. It's been fun.
This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form and you can mix mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.